Hi students, let us discuss further about the transcription process in eukaryotes. I mentioned already it is somewhat complicated. We have the gene, the split gene, having the exons and introns. And also, we have some processing. One, the splicing, that's a common one, splicing process. And two, additional processing. One, what is called the capping process, another one, the tailing process. These are all the things what happen just in the case of eukaryotes. I discussed earlier, but without this diagram, somewhat a different diagram. The same one I want to repeat here. You see that one, the RNA polymerase 2 is responsible for messenger RNA. The mature mRNA is being formed only by the action of RNA polymerase. Now this RNA polymerase binds to, that is what is called the promoter region. There we have the Tata box, which directs the RNA polymerase to the site of transcription, gene transcription, or gene what we have, actually the message is being transcribed in mRNA. Now, the RNA is being synthesized in the direction of 5' 3'. Now, the template strand for this one in the DNA is running 3' 5'. See that one, 3' 5'. Now the RNA is being synthesized with the help of RNA polymerase 2 and subunits, some are present inside, nearly 5 subunits inside the RNA polymerase. Now during the process, two events, one event is what is called the capping. That is nothing but the addition of 7-methyl guanosine triphosphate to the 5' end. I mentioned about the role also. That is addition of, that is 7-methyl guanosine triphosphate is called the capping process. So the capping process is going on along with what is called the elongation of the RNA. So as the gene is split in nature, in DNA, the mRNA which is being synthesized is also split in nature, having both exons and introns. I represent what we call the exon in black color and the intron in green color. So it is being transcribed. Now we have the addition of what is called the 7-methyl guanosine triphosphate. It's a capping process, important for that one, protecting that one, and also for recognition by the ribosomes. Now while the process is going on, what we have, the splicing occurs. The splicing is nothing but a process of removal of introns and joining of exons. At the same time, while splicing is going on, we have the addition of just 200 to 300, what is called this adenosine nucleotides, and the 3' end, and that tail is called poly A tail. The process is called tailing process. Now as soon as what we have the tailing process, and then what we have actually the capping process, along with the splicing completed, we have received what is called the mature mRNA. Now with exons and introns we have premature mRNA. Once the introns having spliced out, we have the mature mRNA having the cap, that is 7-methyl guanosine triphosphate, and a tail with many adenylate residues. So the process of addition of what is called adenine residues is called polyadenylation, 200 to 300. Adenylate residues are added. Now we have the mature mRNA. Now we see represent small circles on the surface of intron. These are all the structures responsible for the removal of introns. They are called spliceosomes, having that one enzyme for the responsible for the removal of introns from that one. So that's called the spliceosome. I represented here the structure made up of RNA and protein responsible for the removal of introns. So the circle represents what is the spliceosome concerned with the removal. So at the end we have received what is called a mature mRNA. The mature mRNA is having at one end what we have the cap, another end what we have that is tail, and also we have some other regions, what is called UTR. So we have UTR at the 5' end and also UTR at what is called the 3' end. We'll talk about what is called the UTR, untranslated regions, later during the process of translation. So this is about the description of what is called the transcription eukaryotes with reference to the diagram as given in the book also. The split gene feature of eukaryotic gene is completely absent in the case of prokaryotes. So in the case of eukaryotes, you know that one, each exon is coded for a single polypeptide or a protein with a definite function. Normally, as we have the split gene, the exon arrangement and the removal of introns is fairly flexible. As a result, the exons coding for these polypeptides, that is the polypeptide subunits, actually become an active domain. The exons act as a domain, combining in different ways to form a new gene. So each axon is acting as a domain. They are combining together in different ways to form a new gene. Normally, a single gene produces different proteins through one process. The name of the process is alternate splicing. Through this mechanism, a single gene produces various types of proteins. So this is the mechanism through which different proteins with a specific function and also the diversity, that is, we can say the functional diversity in animals can be produced. So the main important function of this alternate splicing, this is a mechanism responsible for generating, that is, a functional proteins and also the functional diversity among the organisms. Now, what do you mean by alternate splicing? So it is a process in which the premature mRNA splice themselves in various different ways. The premature mRNA splice themselves in various different ways to produce a gene. That gene produces different proteins. Here I represented what we have the DNA, the split gene, having exon, that is intervened by the introns. I represented two, that is actually 3 exons and 2 introns here. Now what is happening during what is called the transcription process? Now this is transcription, copying of mRNA on the template DNA strand. So now as a result of transcription, we have received, that is premature mRNA, that is also having a split gene, both exons and introns. I mentioned already. The unit which is being transcribed and translated is exon. The unit which is transcribed but not translated is called the intron. Now by means of the process what's called splicing, now the introns are removed. We have 3 exons. These 3 exons are actually joined together in several different ways, forming just actually the genes. For example, the exon 1 and exon 2 join together. This is 1. And exon 1 and 3 join together, and the next one exon 2 and exon 3 join together. So we have received actually 3 different types of genes, and these actually mRNA genes we can see. So they are united and combined together, splicing together in different ways. As a result, we have received 3 different what is called the mRNA, and each one is coding for a specific protein. So as a result of what is called a single mRNA, because of the split gene, 3 different types of proteins which are functional can be produced, and this process called alternate splicing. Nothing but the removal of introns, the union of exons in different several ways or various several ways to form a combinations exon 1 and exon 2, exon 1 and exon 3, and exon 2 and exon 3. All combined together. Each one is normally translated to form a specific protein. This process is called alternate splicing. Now there is a question arises. See that one, the introns would have arisen either before the evolution of eukaryotic genes or before the evolution of prokaryotic genes. So this is what we have. So if they just arise late of the eukaryotic genes, how did they enter into the eukaryotic genes? The question is, so the introns would have arisen either before or after the evolution of eukaryotic, either before or after the evolution of eukaryotic genes. I will take in that way. So if they arise late, that is after the eukaryotic genes, how did they enter into the eukaryotic genes? There is one question, this is a one question, the answer would be. Now, the introns represent the antiquity of what is called the ancestral gene. Now the introns acting as actually mobile DNA sequences. The introns acting as mobile DNA sequences. Mobile DNA sequences. Mobile DNA sequences. And they are actually spliced themselves on their own without anybody's help, either out of the target site or into the target site. So the introns are acting like what's called mobile DNA sequences. They splice themselves out of the target site or into the target site by acting. So they are just actually introduced into the target site or moved out of the target site by acting like mobile transposon elements, like acting like mobile transposons, elements like mobile transposons. Mobile transposons like elements. What is transposons? Transposons are mobile genetic elements. They can insert themselves in any part of the DNA, in any species of DNA. They are also called as jumping genes. The transposons are also called jumping genes. Reported in May 1st by Barbara McClintock. Barbara McClintock just observed these mobile genetic elements, what are called the transposons. They are called as the jumping genes. Now here the introns are acting like what we call the mobile transposon-like elements. They are responsible, or they are acting as a mediator in the transfer of genes between the organisms. They are acting as mediators for the transfer of genes between the organisms. We can have the transfer of genes occur in two different ways. I will represent the same one in a picture. Now the introns entered into the eukaryotic genes because they acted as mobile genetic elements, what are called the transposons. So they are responsible for mediating the transfer of genes, that is between the organisms. There are two ways of what we have the gene transfer. It may be horizontal gene transfer or lateral gene transfer. And the second one, vertical gene transfer. Now what do you mean by vertical gene transfer? There is a common one what we have generally as per in the case of monohybrid or dihybrid or Mendelian inheritance. Now here the characters are transmitted from the parents to the F1 generation, F2 generation, F3 generation, so on. This is called vertical gene transfer in a family or in a species, etc. from one generation to another generation of the same species. But here in horizontal gene transfer it occurs between two different species. Okay, for example, this is normally called horizontal gene transfer, HGT, or lateral gene transfer. For example number 1, here It's responsible for the transfer of genes between the prokaryotes, from one prokaryote to another prokaryote. What is studied in the case of Griffith's transformation experiment, we have one strain, what is called R strain, gets converted to S strain because of the gene transfer. Another method in bacteria, the gene transfer method, is transaction between two different species of what is called actually bacteria, where the DNA is transferred from one bacterial type into another bacterial type. And also we have what is called actually, that is conjugation. Here also we have the fertile factor is transferred from one bacterium to another bacterium through the conjugation tube. So these are all examples for horizontal gene transfer occurring between different species. Or it may occur between a prokaryote to an eukaryote. From prokaryote, for example, bacteria, to that is eukaryote. Or between two eukaryotes. For example, we have the TA classmate, the one which is responsible for converting, that is a bacterium, a transgenic bacterium, to produce a transgenic organism. So it may also transfer the genes from eukaryotes to eukaryotes. So we have horizontal gene transfer occurs either between prokaryote to prokaryote, or between prokaryote to eukaryote, or between two eukaryotes. So as a result of this activity of transposons or introns, now the horizontal gene transfer have known to play a major role in the evolution of life, that is on Earth. So now the evolution of life on Earth depended on what we have, horizontal gene transfer, a method responsible for transferring the gene from one species to another species. That leads the evolution of new forms of life on Earth. Now students, let's pass on to the next part, what we have, the genetic code. We know that DNA is a carrier of genetic information in the cell and also between the organisms. Then we would like to know in what form the message is there in the genetic material. That is an answer we have to find out. Now in the case of translation process, one of the events, the second event in the gene expression, we have a sequence of 3 bases in mRNA, normally called as codon, which designate one of the different amino acids, one of the 20 different amino acids which are taking part in protein synthesis. So I can say in nature there are about 20 amino acids. These 20 amino acids are responsible for the formation of proteins by different combinations. They combine together to form a specific protein with a specific function. Now what is a genetic code? Now this is a sequence relationship between the nucleotides of DNA and the amino acids of protein that they are encoded by, that is the DNA. Suppose I am taking the DNA, this is the DNA, one of the DNA is acting as a template strand, now this is template DNA, I am taking only one strand, having the bases like this, this is also the nucleotide sequence. Now this nucleotide sequence is being transcribed in mRNA, so I am taking this is mRNA. Now the bases present in mRNA are complementary to the bases of the template strand. If you have A, here you have T, A, C, then G, U, G, that is A, U, then C. So this is the base sequence in mRNA. They represent the codon. Then this is what we have, the transcription process. Transcription, the message is transcribed. Then which undergoes a process of translation to form a protein. And we have a number of amino acids like this. So one amino acid I am taking. So the genetic code is the relationship between the nucleotide sequence of DNA and the amino acid sequence of protein, which are normally that is encoded by the DNA. This is one. What is the genetic code? In nature, in living organisms, there are 64 codons. 64 codons. What are codons? A set of 3 nucleotides, a set of 3 bases carried by mRNA. A set of 3 bases carried by mRNA. Suppose I am taking this is mRNA. I am taking just a set of 3 bases. They carry the message for one amino acid. For example, if you have this is mRNA, this is a code that is normally coding for the amino acid methionine. Methionine. Okay. So there are 64 codons for 20 different amino acids. So total number of codons 64. Total number of amino acids. Total number of amino acids, that is we have 20. That means we have more than one codon for an amino acid. More than one codon for an amino acid. Now 64 codons I mentioned. Out of these 64 codons, only 61 codons specify the amino acids. 61 codons specify the amino acids. That means they have the message for the amino acids. The remaining 3 are responsible for the termination signals for a polypeptide chain. When to stop the process of transcription after the transcription is completed for a particular polypeptide chain. These 3 that is codons are called actually the termination codons because they provided the termination signals for the polypeptide chain to be completed after transcription process. Now, as we have 20 different amino acids, the meaning for that one, there is more than one codon for each amino acid. There is more than one codon for each amino acid because the number of codon is more when compared to the receptor. So the meaning for this one, this actually the property is called degeneracy of codons. This property is called degeneration of codons. Or we can see the codons are degenerate in nature. Codons are degenerate. The codons are degenerate because we have more number of codons than the amino acids. That is why we have there is more than one codon for an amino acid. That phenomenon is called degeneracy or the codons are degenerate. What are the reasons for the multiple nature of codons for an amino acid? That is what we call the degenerate codes. The multiple nature of codons for an amino acid. That is what is called the degeneracy. Now there are 2 reasons given. There are 2 reasons given. Reason number 1. What are the reasons for the multiple nature of codons? Reasons for multiple nature of codons. Multiple nature of codons. Reason number 1. There are more number of tRNAs for amino acids. So the number of tRNAs, we can say this way, number of tRNAs are more than that of the number of amino acids. Because the tRNAs are the carrier molecule. Adapted molecule, they only brings the amino acid, carrying the amino acid towards what is called actually the site of protein synthesis. Again we have, as we have many tRNAs, now there is a set of 3 nucleotides in mRNA carrying the message for one amino acid is called as a codon. Codon is nothing but a set of 3 nucleotides. I will talk about it later. A set of 3 nucleotides carried by mRNA having the message for one amino acid. The message is read by another set of 3 nucleotides present in tRNA. That is called anticodon. A set of 3 nucleotides found in tRNA which are complementary and read the message of mRNA. They are called actually that is anticodons. So each tRNA or simply the tRNAs have, we can say that tRNAs have different anticodon. That means the anticodon of one tRNA is different from another anticodon of another tRNA. This is reason number one for multiple nature of codon. The second one, the pairing between codons and anticodons. That's the second point. Pairing between codons of mRNA and anticodons of tRNA. Is highly specific. So that what is happening? Highly specific, particularly between the first 2 positions on the codon, highly specific, that is for the first 2 positions on codon. Now this one permitting that is Watson and Crick base pairs to be formed. This is the second reason. What is the meaning for that one? Suppose I am taking a strand of mRNA with some codons. So this is mRNA having one codon UAC, and this is for amino acid tyrosine. To read this message, if you are taking the tRNA, it has a set of 3 nucleotides. This is tRNA. That is A, that is U, G. So this is what is called the codon carried by mRNA. A set of 3 nucleotides carried by mRNA. Now this is what is called the codon. A set of 3 nucleotides carried by tRNA is called anticodon. The bases between mR and tRNA are complementary to each other. Base pairing rule. They are following the base pairing rule. That's why I mentioned about. So they follow what's called permitting Watson and Crick base pairs to be formed. So base pairing occurs. Now the first 2, the bases are highly specific. So here there is no change. If you have U in mRNA, definitely in tRNA you have A. If you have A in the second position, that is in mRNA, you have definitely U. So the first 2 positions in what is called the pairing between mRNA and tRNA, we can say either in codon or anticodon are highly specific. Whereas the third position, see that one pairing between codons of mRNA and anticodons of tRNA is highly specific. That is the pairing. For the first 2 positions, 1, 2, there is no change. If you have A, definitely A is there in anticodon. On codon, permitting Watson and Crick base pairs to be formed. So the base pair is formed based on Watson-Crick model of base pair. But in the third position, if you are taking the third position, what will happen? So the base pair at the third position, there is a great flexibility as to which base pairs are acceptable. That's what I mentioned in the case of the third position. The first two positions are highly specific, but the third position is highly flexible. And normally we have a hypothesis, what is called wobble hypothesis. Wobble hypothesis. I will discuss later. Now the third base, that is in the case of anticodon, is called as wobble position. Wobble position. Plain one, highly flexible. The meaning for that one, highly flexible. And the hypothesis proposed is called wobble hypothesis. Okay, now normally you see that when the genetic codes are universal in nature. We will discuss later the properties. Now, the order of just base pairs in a DNA molecule controls the kind and order of amino acids in a protein. So this specific base pair is called what is known as a genetic code. The specific base pair is called actually genetic code. The one which decides the sequence of amino acids in a protein. Now several people have contributed for the disappearing of genetic code. Let's see some of the persons who contributed for the disappearance of genetic code. Open up, disclosed. Number 1, George Gamow. So he actually proposed that a set of 3 nucleotides must be needed to code for 20 different proteins. A set of 3 nucleotides must be needed for coding 20 different proteins. Number 2, Marshall Nirenberg. (1961). He was the man disappeared the first genetic code. Disappeared the first genetic code. Disappeared the first genetic code. The first genetic code that was disappeared is U. This is the first genetic code for an amino acid phenylalanine. So this is the first genetic code disappeared by Marshall Nirenberg in 1961, and that was U for phenylalanine. Now, third person, that is Severo Ochoa. Severo Ochoa. Now, he just actually used a method, what is called enzymatic method of RNA. Enzymatic synthesis. Enzymatic synthesis of RNA. He used the enzyme polynucleotide phosphorylase. The name of the enzyme, polynucleotide phosphorylase. This enzyme is also called as Ochoa enzyme. So he used actually enzymatic method of synthesis of what we have RNA. Then the next one, Indian born American Hargobind Khurana. Hargobind Khurana. So he used chemical methods for the synthesis of RNA. So these were the different people. Marshall Nirenberg, Severo Ochoa, polynucleotide phosphorylase enzyme, that is called the Ochoa enzyme, and also Hargobind Khurana just use the chemical methods for the synthesis of RNA. Now these are all the different people who have, and because of their work, Nirenberg, Hargobind Khurana, and also Hurley, the one who proposed the cloverleaf model of tRNA, along with what we have anticodon. So, with Corona, Nierenberg and Hurley were awarded a Nobel Prize for their contribution to genetic code and also for anticodon and structure of tRNA, etc. So, these were the persons normally just contributed towards the genetic code, and mainly Nierenberg, he was the first person disappeared, opened up and disclosed, that is the first codon, the genetic code for phenylalanine table U. Now just I would like to say only one statement, that is the sequence in mRNA. So the sequence in which the mRNA has the basis will design the sequence of amino acids in a protein. Okay, the sequence in mRNA, that is the basis, which will design the sequence of amino acids in a protein. Now we have to go further about the properties of genetic code in the next class.